So this is our sixth BioNMR workshop, and the topic is on variable temperature NMR. So first, let me introduce our BioNMR staff, and Deb and I are responsible for the 600 megahertz and 800 megahertz NMR located in the chemistry building, and there is Sukhana. And Emily is our faculty director, and Anne is the director of operations. And starting 2 years ago, we started to hold regularly those BioNMR core workshops. And so far, there are 5 workshops that are available on our BioNMR core website. And those video links are listed here. And we've done workshops on 1D and 2D NMR methods for small molecules, protein backbone dynamics. And there are 2 workshops on protein backbone assignments, one on using Sparky to do the assignments, and the other on using CCP AMR analysis assign. And the latest one is done by Deb on tips and tricks for collecting good AMR data. And for this workshop today, it is being recorded. Later on, we will share it on our BioNMR website, so you can watch this video anytime. And webcams are optional for hosts and participants. And if you are given the opportunity to be on stage, please watch if your microphone is mute or not. And also feel free to use the Q&A panel or raise hand to ask questions. And feel free to ask questions at any time. And then we will move on to our topic on variable temperature NMR. So this workshop will have 5 parts. And first, we will give some example on why we want to run variable temperature NMR experiments. So we will learn the applications of variable temperature NMR experiments, both for small molecules and proteins. And then once you decided you want to run variable temperature NMR, I will cover the variable temperature stabilities, capabilities of each of our NMR instruments, so you know which NMR instrument to choose. We will then cover sample preparation tips, experimental setup, and finally, we will have a summary session on important considerations for VT NMR. So if you have not run variable temperature NMR experiments before, the first question you may have is why you want to run these kind of experiments and what kind of information can you get from these kind of experiments. So variable temperature NMR experiments, they are defined as experiments that are done at temperatures higher or lower than 25 degrees Celsius or 298 Kelvin. And here I give some examples of the applications of VT NMR experiments. So it was used most commonly for conformational or chemical exchange studies. And also, if you want to follow a high or low temperature reaction, you can run VT NMR experiments to check your reaction. Or if your compound is not very soluble in the solvent you choose, then you can increase the temperature to help with the solubility. Also, if your sample is not very stable at room temperature, then you can decrease the temperature to help stabilize your sample. So we just go through those examples one by one. So the most common application is to use it to study chemical exchange or conformational exchange. And the example I give you here is a very classic example. This molecule is an N-dimethylacetamide, and it has 2 methyl groups attached to this tertiary amide bond. And also it has another methyl group attached to the carbonyl carbon. So at first glance, you may think that this molecule for this methyl group A and B, they are chemically equivalent because it seems this tertiary amide bond can rotate freely. So then you can record a 1D proton spectrum of this molecule at room temperature. So that is how the spectrum looks like. So from the spectrum, we can see that A and B are not chemically equivalent, and their chemical shift is different in the 1D proton spectrum. So why is that? And this is actually because this amide tertiary bond does not rotate freely. So it has those partial double bond character. So this A and B methyl group, they can be considered sitting cis or trans to this carbonyl oxygen. So they are not chemically equivalent. And then to confirm this that the A and B are not chemically equivalent, you can actually change the temperature and see how your spectrum changes as you increase the temperature. So if we increase the temperature to 350 Kelvin, now you can see that A and B peaks are getting more broad and they start to merge into one peak. And if you increase the temperature a little bit more, then these two peaks eventually merge into one very sharp peak. And this temperature eventually went to 420 Kelvin. So why do we observe those changes in the peaks as we increase the temperature? So we need to understand exchange regimes. And so in the example I gave you, this is a very simple case that we have two states or two sites, A and B. They have equal population and they are under exchange, and the exchange rate is K. So at low temperature, these two states or these two sites A and B, they will show two peaks in the 1D proton spectrum. And we call that at this temperature, A and B undergo slow exchange, meaning that the exchange rate is slow on the AMR chemical shift time scale. So that means the exchange rate K is much smaller than the chemical shift difference between these two states. So the lifetime of each of the states A and B are so long that AMR can distinguish between these two states. So all signals of components are observed. And then as we increase temperature, we can reach a coalescence temperature. So at this temperature, these two states, these two peaks merge into one broad peak, and we call this intermediate exchange. So under intermediate exchange, the exchange rate is comparable to the chemical shift difference between these two states. So two signals merge into one broad peak. And at high temperature, what we see is that the two peaks merge into one sharp peak. And this is called fast exchange. And when the temperature is high, the exchange rate actually becomes very fast. So it's faster than the AMR chemical shift time scale. So exchange rate K is much larger than the chemical shift difference between the two states. So the lifetime of each of the states A and B are so short, so AMR cannot distinguish between these two states. So now you see just one cross peak, so a single resonance is observed. So we can actually analyze these chemical shift changes and also the line shift changes of under slow exchange, intermediate exchange, and fast exchange. And then we can derive some important parameters like rate constant and activation energy. And these kind of studies are called dynamic AMR, and they actually have a wide applications. So here I give you an example which is very similar to the example I gave you before, that these two groups A and B are attached to a tertiary amide bond. So A and B are not chemically equivalent. And under a restricted amide rotation, we can actually exchange. So in this example, the authors recorded the 1D spectrum of this molecule at increasing temperature. So from there, they can overlay the spectrum and they identified slow equilibrium, coalescence temperature, and fast equilibrium. And then from here, they can actually derive the important parameters. I give you an example here is they can actually calculate the rate constant at the coalescence point. So you just need to put in the chemical shift difference between these two states, and then you can get the rate constant. Also, you can calculate the activation energy. So you also can calculate through this equation over here. So they were able to calculate the energy barrier for the restricted boundary rotation to be 18.4 kcal per mole. And also sometimes using VT NMR, it can help with improving resolution. And here's an example that this molecule is sucrose. It is dissolved in DMSO-D6. And all those hydroxyl group actually will appear in your 1D spectrum. And together with those proton groups in your ring system, then this 1D proton spectrum will be a little bit complicated. So then you can actually increase the temperature and see how the chemical shift of those peak changes. So especially if you pay attention to the peaks H32, as you increase the temperature, they actually get separated more and the resolution get improved. So this is because as you increase the temperature, the intermolecular hydrogen bond get affected, and also the population of each of the exchange states get affected. So that results in the improved resolution. So if you have a molecule that its structure is complicated, and then you may try to change the temperature to see if the resolution get improved, and that will help you with peak picking and integration and so on. And VT NMR can be used for reaction monitoring. This is a very straightforward example here that you can put your reactants in the AMR tube and you record the 1D spectrum of the reactants. And then as you increase the temperature, you can then monitor the changes of the 1D spectrum. And that will tell you if you get the product or not. So in this example, as we increase the temperature, we can see that the products slowly appear, and then you can select the most appropriate temperature for the reaction. And this is an example for when using VT NMR to do solubility studies. So as you change the temperature, of course, the solubility of the compound will change. And in this example, the authors, they study the structure of this molecule called HSA. And then they record the 1D proton spectrum of this molecule and also at increasing temperature. And then they monitor the intensity of each of those peaks. So at low temperature, the intensity of the peak is low. And as you increase the temperature, the intensity increase and eventually reach a plateau. That means the solubility is the highest at this specific temperature. So they were able to determine the solubility of this compound using this kind of experiment. And finally, these VT NMR experiments can be applied to protein structure studies. And this example that the authors use these VT NMR experiments to study the stability of the protein. So on the left is a proton nitrogen HSQC spectrum of this protein. And if you haven't run those kind of experiments before, it's actually very similar to a proton carbon-13 HSQC spectrum. So now on the y-axis, we have nitrogen-15 chemical shift instead of carbon-13. And then each of these cross peak indicates a correlation between the backbone amide proton and its directly attached nitrogen. So each peak can represent one residue in your spectrum, so the backbone of that residue. So, and then what the authors did is that they increased the temperature from 5 degrees Celsius to 45 degrees Celsius, and then monitor the chemical shift changes of each of these cross peak. And the larger the change, meaning that this residue is less stable, and the smaller the change, then that means this residue is more stable. And then you can actually quantify those changes and then map that chemical shift change to the structure of your protein. And then that can be used to study the local structural stability of your protein. Okay, so that is for the some examples of the applications of variable temperature NMR instruments, variable temperature experiments. And then once we decided to run these VT NMR experiments, we can choose the appropriate instrument to run this experiment. So we'll go over this and then you know which NMR instrument to choose. So first, let's take a look at how the sample temperature is controlled inside the NMR coil or inside the NMR probe. So your sample sits inside the probe, and on the outside of your sample is this NMR coil that detects your signal. And below this sample, you can see there's a pipe over here. So it's actually the variable temperature gas that flows through this pipe and then flows through your sample from the bottom to the top and then leaves at the top of the probe. So your sample will get heated or cooled by this VT gas flow. And then at the bottom of your sample, there's also a temperature sensor that actually detects the temperature of the VT gas. And inside the pipe, there's a heater that heats the your variable temperature gas. And then also there's an external chiller that provides the cooling gas to cool your sample. And we always want to provide this flush gas. And this flush gas is used to prevent damage to your probe. So when you are doing some heating experiments, it can prevent overheating. And if you are trying to cool your sample, this will prevent icing. So that's how your sample is cooled. And then this is a table that compares the variable temperature capabilities of each of our instruments. And so the temperature range of each of the instruments is determined by the probe and also the cooling device. And for the 600 megahertz AMR in Marysville Common Hall, it has a nitrogen-cooled probe, and we have an FTS chiller that provides the cooling gas. And the temperature for this instrument range is 5 degrees to 171 degrees Celsius. And we don't recommend going lower than 5 degrees Celsius. And because in our previous experience, while the temperature is lower than 5, there may be icing issue. So this lower limit is 5 degrees Celsius. For the instrument in 600 megahertz and 800 megahertz in KM1047, they are both helium cooled, and they both use a Bruker Smart Cooler BCU1 to provide the cooling gas. And here are this temperature range of these two instruments. So you can just check the temperature range here and then compare with your target temperature, then you can get the appropriate instrument to choose. So we just take a look at different the chillers and BCU unit in different instruments. So this is a picture of the AMR in Marysville Common Hall, and each of the part of the instruments are labeled. And this chiller is actually located at this corner over here. You may not even notice that. And that's the FTS chiller for this instrument. And it's down here on the corner and next to the console. And on this FTS chiller, it provides the cooling gas, and the temperature of the cooling gas is actually displayed on the screen here. And then it has a hose that transfer those cooling gas and then to the probe. So on that FTS chiller, there's a power switch. So this power switch first needs to be turned on so that the FTS chiller can work. And then on the top display, it actually displays the actual variable temperature gas temperature. And then on the second display, it actually displays the desired VT gas temperature. So you can set your desired VT gas temperature by pressing this up and down arrow. And usually you want to set it 10 degrees Celsius lower than your desired temperature. And for the 600 in KM1047, the 600, its BCU unit is actually located on this right corner. And it's over here. This is called the BCU1 and provide cooling gas to the probe. The 800 has a similar or the same BCU chiller located on the very left, and it's located here, also provided the cooling gas to the probe. And on that BCU chiller, you can see there's a rotary switch. So it can be rotated to remote operation mode or manual operation mode. And if it's under manual operation mode, you can choose 0, meaning there's no cooling power, or 1, that is for medium cooling power, or 2 for maximum cooling power. And if you switch to the remote operation mode, then on top spin, you can actually remotely control this chiller. So I will show you later on how to control that. So you can actually press the set button here, and then that can increase or decrease the power of this chiller. Finally, there are some very useful commands that I think will be useful to use for you to check the temperature range of each probe. So the first command is called add probe, EDPROBE. So you can just put this command into the top spin command line, and that will open up this add probe window. And here you can select the probe that you want to view the properties. So we only have one probe for this in Marysville Common Hall and 600. So we select the probe, click on view properties, and then you go to temperature parameters. And here you can see the minimum permissible temperature and maximum permissible temperature for these types of probe. So that's one way to look at the temperature range for the probe. And another way is to type EDTE later on, we'll use this command very often. So that will enter the temperature control suite. And then you can go to configuration. And here you can also change the unit you want to display for the temperature to Kelvin, degrees Celsius, or Fahrenheit. And also at the bottom, you can see there's a temperature limits that also display the temperature limits for this type of probe. But now this is set in Kelvin, not in degrees Celsius. All right, so that is for the variable temperature capabilities of each of our instruments. So I'll just wait to see if you have any questions. So you can either type it in Q&A panel or raise hand. And if there's not any questions, we'll just move on to sample preparation. So there are some very important considerations that we need to take when running those variable temperature NMR for sample preparation. So the first thing is for selecting the appropriate solvent. So when running the variable temperature NMR experiments, you want to make sure that your solvent is stable at the target temperature. So you want to get the melting point and the boiling point of your sample. And here I just listed the melting point and the boiling point of the most commonly used solvents. For example, if you want to run high temperature NMR experiments, then DMSO will be a good choice because it has a high boiling point. And if you want to run low temperature experiments, then choose something like acetone, acetonitrile, or methanol, which have low melting points. So the general rule is that you want the highest temperature allowed should be at least 10 degrees Celsius below the boiling point, and the lowest temperature should be at least 10 degrees Celsius above the melting point. And also here I put in a link for the data chart. So this is a data chart from Cambridge Isotope that it gives the properties of almost every NMR solvent. And from there, you can also get the melting point and boiling point of your solvent. And the second consideration is for NMR tube. We don't want to use those cheap economy NMR tube. Instead, you want to use those higher quality precision NMR tube. So I'm using Wilmette lab glass tube as an example. So you want to choose those precision tubes, and also you want to look at the field allowed by those tubes. So for our 600 and 800 megahertz NMR, you want to choose those 535 pp7 NMR tube for your variable temperature NMR experiments. Those economy tubes, they are cheaper, but they may deform or fracture at temperature extremes. So they are not recommended for VT NMR. And next, we also want to think about sample stability. So once your samples are put into the NMR tube, you want to make sure that your sample is stable at your target temperature. Also, we don't want to seal the tube. So if the tube is sealed, then that may result in an explosion. So you just need to use a regular cap to just cover the top of your NMR tube, then you are good to go. And sample volume is a very important consideration point while running variable temperature NMR experiments. So let's take an example that if your sample is heated, and as I showed you before, it's heated by the VT gas and it's heated from the bottom to the top. So the lower part of your sample will have higher temperature and the top of your sample will have lower temperature. And that temperature variation across the sample can lead to convection. And if convection exists, then that can result in bad shimming and broad peaks. So to prevent convection, we can use shorter sample. So my recommendation is to use 400 microliter sample. And if you have less than 400 microliter, you may have shimming problem. If you have more than 600 microliter, that can greatly increase temperature gradient and result in convection and also bad shimming. So in our previous workshop on tips and tricks for collecting good NMR data in the advanced shimming routines part, and Deb actually introduced the command called convection, COMP, convection compensate. So this command in TopShim can compensate for convection currents at higher temperatures and large sample volumes. So if you're interested in how to use this command, you can go back to that previous workshop and watch this part. And finally, once your sample is ready, we need to choose the correct spinner. There are 3 types of spinners that are available from Bruker. And the most commonly used one, and most of you are familiar with, are the blue spinner. And it can be used for temperatures in the range of 0 to 80 degrees Celsius. And if you want to go higher or lower temperature, you can change to the KLF or ceramic. The ceramic spinners are good for almost all the temperature range from negative 150 to positive 150 degrees Celsius. Okay, once the sample is ready, then we can set up the experiment. This part, I will include how to establish temperature. So I'll show you how to establish high temperature and low temperature manually. And then we also cover how to monitor and record temperature. Then a little bit on temperature calibration, and then finally a few slides on how to do those VT NMR experiments using Icon AMR automation. So the temperature can be established through the temperature control suite. This is an example from the 600 in Marisu Common Hall. So you can just type in EDTE in the command line, and that will open up the temperature control suite. So first, you can just go to the temperature tab. Under the temperature tab, it will show you the current variable temperature state. So first, you want to make sure the VTU state is on, and then you look at the channel that you are interested in, and then the regulation state should be steady. The stability part will tell you when it reaches stability. So here, your sample temperature is displayed. It should be green, showing meaning your sample is stable. It also has this function to set the target temperature and also show you the heater power. And the next line shows you the probe gas flow. So that is the gas flow for the VT gas that is used to cool or heat your sample. And usually we just set it to 535 liter per hour. So you don't need to change this number. And to run high temperature AMR experiments, you can just directly go to this temperature setup and just click on set. And here you can enter your target temperature. Once you entered your target temperature, for example, here I entered 305, and then you can just watch how the temperature changes. So if initially your temperature is lower than the target temperature, so your sample temperature is shown in blue, and then the heater will turn on, so the heater power increase. And then the temperature will go up a little bit above your target temperature. So now the temperature is showing in red. And at this point, the heater power will start to decrease. And eventually, the sample will reach a stable target temperature at 305 Kelvin. And now you see the regulation state is steady, and it also shows you the time that it reached the stability. And to run low temperature experiments, the first thing you do is to change the temperature on the FTS chiller or change the power on the BCU unit. So here I will first show you how to do that on the FTS chiller, which we mentioned before. So first you change the desired temperature on the FTS chiller for the 600 in Marisu Common Hall by pressing those buttons. And then you want to set it to 10 degrees Celsius lower than your desired experiment temperature. And now you can go to that temperature control suite and then change your target temperature here. And then you can just wait for your temperature to reach the target temperature. And for the 600, 800 in Chem 47, the display of the temperature control suites are almost the same, except that it has one extra line down here that control the BCU. It's because the BCU is under remote operation mode, so you can actually set the power remotely. So now you can just set the BCU power here. Generally, if you want to run temperatures higher than 30 degrees Celsius, we set this power to medium. And if you want to run low temperature experiments lower than 10 degrees Celsius, then we'll set this power to max. And I'm just to summarize here some general rules for setting the target temperature, which I also mentioned before, is that you want your highest temperature be 10 degrees Celsius lower than the boiling point of your solvent. And also your lowest temperature should be at least 10 degrees Celsius above the melting point. And while setting up the temperature, you don't want to increase the temperature dramatically. For example, if you want to increase the temperature like 60 degrees Celsius, you want to do it step by step. So each step you increase 10 to 20 degrees Celsius and wait for 5 minutes for the temperature to stabilize, and then increase another 25 degrees Celsius and so on. And this is to prevent damage to the probe. And always comes out with us first if you run in the VT NMR experiments for the first time. Okay, once your temperature is established, then I will show you some useful features on how to monitor and record the temperature. This might be useful if you want to run a very long temperature and you want to make sure the temperature is stable during the whole experiment. So this can be done by just go to the monitoring tab. So under the monitoring tab, you can select the parameters you want to look at. Most often, I just select temperature and target temperature. You can also look at other parameters like the heater power, gas flow, and target gas flow. So here, I increase the temperature to 305 at this time point. So now this target temperature immediately increased to 305. And then the sample temperature will just slowly go up and go above 305. And then eventually it reached the target temperature. And if you don't want to sit in front of the computer and watching this temperature all the time, you can actually go to the record tab. Here you can choose to record the temperature. So first you want to choose the recording intervals. So it can be 10 seconds, 1 minute, or 1 hour. And then you also put it, all the temperature data will be stored in this file. And then you can select the parameters you want to monitor, like temperature and target temperature. And now just press the start button, and then the system automatically will record the temperature. The temperature data will be stored here. Once your experiments are done, you can go to show and then select the file that stored your temperature data. And then it will show you the graph of your temperature and target temperature over the entire experiment time. Okay, sometimes we also need to do temperature calibration. And this is because that the temperature that you measured from the temperature sensor is not the same as the temperature of your sample. So this is actually a picture of the probe. So your sample sits in the probe, and the sensor, which is this red dot over here, it's at the bottom of your probe, and it measures the temperature bottom of your sample, and it measures the temperature of the VT gas, not the temperature of your sample. So the actual temperature of your sample can be a few degrees different from your sensor temperature. And if you want to know the actual temperature of your sample, you need to calibrate the temperature. So we do those calibration using some calibration samples. And here are some examples. There are other calibration samples you can use. And the example I give here is that for low temperature, you use 100% methanol for temperature calibration. And room temperature or a little bit above room temperature, you can use methanol D6. And for high temperature, you can use 100% ethylene glycol for temperature calibration. And here shows we how we do the temperature calibration. This is a ethylene glycol. So what you do here is that you record the 1D proton spectrum of this calibration sample, ethylene glycol, at increasing temperature. And then for each of these spectrum, for example, for this spectrum at 303 Kelvin, you measure the chemical shift difference between this OH peak and the CH2 peak. And then you put this chemical shift difference into this equation down here. Now then you can get to the actual temperature of your sample. So for each temperature, you can put in this chemical shift difference and get the actual temperature for each of the sample. And these are the example I showed you here is to do it manually. And now on Bruker top spin, you can actually do those calibration automatically using a command called caltemp. So you acquire that 1D spectrum and then type in caltemp and then select the sample you are using. For example, here I can select the methanol D6, and then there will be a pop-up window showing up that it automatically measures the chemical shift difference, and then it calculates the actual temperature of your sample. And if we don't want to do the calibration every time while you are running the sample, then we can actually first build a calibration curve and then use that curve for future calibration. So this kind of temperature calibration curve have your sample temperature on the y-axis and the sensor temperature on the x-axis. So you just draw a curve of this sample temperature versus the sensor temperature. And then next time when you want to run a sample at a specific temperature, you just need to put in the sensor temperature and then you can get the sample temperature. So in top spin, we can also get that kind of calibration curve automatically. So you can just go to correction and then here select the probe you are using and then enter the temperature that you get from the calibration sample. And automatically, this correction system will calculate the calibration curve. So it will put a slope here and an offset here. So that's the slope and offset for the calibration curve. And then in your future experiment, all those sensor temperature will be calibrated to show you the actual temperature of your sample. So that is why on the display of your sample, temperature display of your sample, it always say corrected temperature. So the sensor temperature is actually at 298.5 Kelvin. And after calibration through that calibration curve, then we can get the corrected temperature, which is 298 Kelvin. There's another very useful feature on the temperature control suite called self-tune. And this self-tune can be used if when you're trying to reach a target temperature, you find that the temperatures are oscillating, the temperatures are not stable, or it takes too long to reach that final temperature. Then you can actually do a self-tune to optimize some parameters that control the temperature. So for example, here you can, once you reach the target temperature, you can just press start and then stop to do the self-tune. And the self-tune will automatically optimize several parameters related to sample control. As I show here, these are parameters that it's trying to optimize. Once those parameters are optimized, then the temperature will get better regulated, and that will solve those temperature oscillating issues. And also you can store those self-tune parameters for future use, and you can also view those parameters. All right, finally, we'll just go through how to set up those automations using Icon AMR. And I'm also using the 600 megahertz in Marisukamma as an example. And I recommend that just use Icon AMR for temperature range from 5 degrees Celsius to 80 degrees Celsius. And during this, at this temperature range, you don't need to change to other spinner. So you can just use a blue spinner for your experiment. But if you're going higher than 80 degrees Celsius, we need to change to that ceramic spinner. And this spinner is much heavier than the blue spinner. So we have to manually adjust the ejection airflow to eject the ceramic spinner. So this is not very convenient to do under Icon AMR. So if you're going higher than 80 degrees Celsius, we will do manually. And in this range, we'll do it in Icon AMR. The 600, 800 in the Temp 47 may have a different setup. So you need to consult with Deb to see if you can do that through Icon AMR. So setting up those VT NMR experiments is very straightforward. So you log into Icon AMR and you need to select the experiment you want to run. And on this drop-down menu, I put in 2 experiments that is designed specifically for VT NMR experiments. So they are about the same as regular 1D proton and carbon-13 NMR experiments, except that we add about 5 minutes equilibrium time to allow the target temperature to stabilize. And after that 5-minute equilibrium time, then it will do shimming and acquisition. So you can just select those two experiments. And then under the parameter tab, you can select the temperature you want to run. So just set the target temperature over here. Also, as I mentioned, that you don't want to increase or decrease the temperature dramatically, and we want to do it incrementally. So we can set up a series of VT NMR experiments with 20 Kelvin increment. So, and each of these, you can set the temperature over here. And then just click on submit, and then Icon NMR will automatically run those experiments and then send you the data by email. All right, so that's all for those variable temperature NMR experiments. We'll just have a summary session on important considerations for VT NMR. And first is before you start the experiment, make sure you check the temperature range of the instrument, use the right solvent, select the appropriate spinner, and make sure your compound is stable at the target temperature. And also during the experiment, we want to approach the extreme temperature slowly. And also we can calibrate the temperature and do self-tune if needed. Finally, this is a workflow for running VT NMR manually. This summarizes what we just go through in these slides. So first we open the temperature control suite using EDTE. And depending on the target temperature you want to set, you may want to change the FTS chiller temperature or change the BCU mode. And then we'll just set the target temperature. Make sure you set it incrementally, so 10 to 25 degrees Celsius for about every 5 minutes. And once the temperature gets stabilized, you can acquire the spectrum. And when that is done, we'll finally reset the FTS chiller and BCU to the original value. So we also want to return to the room temperature slowly. And for this part, you can just do it in 10 to 20 degrees Celsius for about every 1 minute. Okay, so that's all for our workshop on VT NMR.